Hi, and welcome to part 3 of the Forensic DNA Profiling Lab. My name is Jeffry Petracca, and I'm an educator here at the DNA Learning Center at Cold Spring Harbor Laboratory. Now, you have already joined us for part 1 and part 2 of this experiment. You will know that we have extracted DNA from cheek cells in 3 samples, actually. And then we last time took a look at the gel electrophoresis results of running a quick gel, agarose gel, in order to see whether or not our amplification with PCR was successful, and to kind of get an idea of what genotype we might be expecting at the D1S80 locus. Now, for this final part of the experiment, we are going to use the Agilent Bioanalyzer to run a DNA chip, which is very much like gel electrophoresis on a smaller and more precise scale. We will go through running that chip, then we'll talk a little bit about the meaning of the results. So let's go on up to the board. And so just a reminder of the purpose of this lab. It was to identify the number of repeats, or essentially, more generally speaking, the genotype at the D1S80 locus on chromosome 1 for 2 of my samples, as well as another unrelated sample. And so we did this last time with gel electrophoresis, which, as we saw the results, while it did show us that the amplification was successful, it did not give us a good way to tell the exact size and the exact number of repeats that we have at the D1S80 locus. So the amplicons from PCR, that is, we were not able to identify the precise number of repeats. So this time we are going to visualize our DNA a different way using a DNA chip. Now, the DNA chip works in a very similar way to gel electrophoresis. So up on the board here, you can see an image of one of those chips. And if you look carefully on the chip, you'll notice these little sort of matrixy looking computeristic little features. And these are known as microfluidic channels. And essentially what winds up happening is the DNA will run through the chip through these microfluidic channels through a gel matrix, believe it or not. It just won't be an agarose gel matrix. It'll be a slightly different, more refined polysaccharide matrix. And the DNA itself will actually be pushed through this gel and will be interpreted by a laser in a machine known as a bioanalyzer. And what this will allow for is a more precise resolution and a more precise determination of the exact number of base pairs that make up a particular amplicon, a particular piece of DNA going through this DNA chip. And so essentially what that allows for is higher accuracy, and it will be reproducible as well. And so the benefit of this, besides those things, is that it also requires a minimal amount of sample. So you guys saw how in the gel electrophoresis component of our presentation, we use about 15 microliters of our sample all in all. Now, if we use a small number of sample, we could probably use a smaller number of amount of sample than that, but it may not produce a very clear gel result. That's why we use so much sample. In our DNA chip, only 1 microliter of our sample is actually necessary. Now, when you're talking about a crime scene or something like that, you might not have a lot of sample to work with. So something where you don't use as much of it probably is going to be a better option to go with, especially if it is more accurate to begin with. And so the only downside to the chip is that it is very expensive and time consuming. So it is a lot more expensive. The primers and the reagents that are necessary to actually load the chip do cost thousands of dollars. And it's definitely much more costly than it would be to run a simple agarose gel, which we're talking like many times the cost involved. And so that's the one downside of the chip. And it is a little bit finicky as well. And hopefully when we run our chip, we'll have some good results, but it can present some issues. But the results that you see from the chip are much different than what you would see from the typical agarose gel. So if you compare on the left here, your typical agarose gel results. So here's our agarose gel. And on the right-hand side, we have the chip results. So we can see very distinct differences here. So for the agarose gel, we can, as we saw last time, we can basically tell the genotype approximately, but we can't actually tell the precise number of repeats or the actual number size of base pairs, the size of the amplicon in terms of the number of base pairs, which is really what we need. On the right-hand side, the results that we get from the DNA chip are something that looks like this. And what essentially this graph up here represents is an electrophoregram. It's an absorbance spectrum. And like I said, what winds up happening is a machine known as a bioanalyzer will interpret the results of the chip with a laser and will measure the amount of absorbance or in fluorescence units. That's what this FU symbol is over here on the y-axis of the graph. And it will compare that to a ladder, much the same way that we did, we used the 100 base pair ladder in the agarose gel. And in this gel up on the board here, this is not a 100 base pair ladder. This is a little bit different type of marker, but the concept is still the same. In our DNA chip, our ladder essentially comes out here and here. These are our reference markers. And so everything that is absorbed in a sample is going to be compared with respect to that reference marker and placed somewhere between the sort of the lower limit of the ladder over here and the upper limit of the ladder over here. So this might correspond to, let's say, 1,500 base pairs, and this might correspond to just 15 base pairs. And so your sample is going to fall somewhere in this area here. The benefit of this is that you'll be able to determine the size of an amplicon right down to a single base pair, or at least plus or minus one base pair, let's say. So sometimes it's not as precise as it could be, but it's definitely much more accurate than a gel would be. And it will even tell you the size of those amplicons down here. It'll give you the number of base pairs that corresponds to each absorbance peak. So here's peak 2 and here's peak 3, and here's peak 2 and here's peak 3 down here. And it'll give you the size of those amplicons that correspond to those peaks. And from that information, you will be able to calculate your amplicon size. Pretty nifty. And this is a very good way to get a precise estimate of the size of your amplicons. And so if we look at this a different way, here is our PCR again. And remember, here's where our primer binds here and here, our left and right primers. And here are all of the repeats inside of our D1S80 region. Remember, there's a little bit of flanking region here and over here that we don't necessarily want to consider. The primers themselves, I should extend that actually, the primers themselves constitute part of that flanking region because those are ultimately all part of the same amplicon. And if we compare it to the results in our DNA chip on the right-hand side, we can see that this larger number of repeats over here, this larger amplicon, which is going to go from here all the way over to here, is going to correspond to that higher peak over there. This smaller amplicon, again, on the other homolog of chromosome 1, the other homolog of D1S80, to be more precise, is going to correspond to that second peak over there. It's a smaller peak. So basically, this graph is kind of like a gel. It's just you're looking at it in profile and you're reading it in terms of absorbance and not so much in the position of the bands themselves. I mean, I guess you are, but you're reading it as position relative to a predetermined ladder, pre-calculated ladder here and here. Now, to actually get the size of your amplicon, we are going to do the reverse of what I showed you in the gel electrophoresis video in part 2. So I told you about this flanking region, and we need to now incorporate that when we are calculating the size or the number of repeats of D1S80 from our chip results. So let's say, for instance, that we got this result over here. Let's say that this was what our data looked like for our amplicon that we loaded into our chip. And so what you're going to see is a couple of numbers. We're going to see peak 2 and peak 3 down here. We're going to have the 426 base pairs here and 517 base pairs here. So the very first thing that we need to do is get rid of this flanking region. Again, we're doing the opposite of what I showed you last time. So 426 minus 149 is going to correspond to a size of D1S80 of 277. Now this is just the repeats, just to be clear. Okay, that's just the repeats. So from this, we can then calculate the actual number of repeats just by simply dividing this by the size of each repeat, which recall, as that number I told you to remember, is 16 nucleotides. So for this allele, we are getting the following result. So 277 divided by 16 for each repeat is going to equal 17.3. which is essentially roughly equal to 17. So that's the number of repeats for that allele anyway. Now we can go through the same calculation for the 517 band. So the 517 band minus the 149 flanking region gives you a total of 368. And that's again just the repeats. From there, to get the size of our the number of our repeats, rather, we're going to do 368 divided by 16, which is going to equal right on the nose 23. So that's the number of repeats for that allele. So therefore, this individual's genotype is going to be 1723. And so there you go. So that's here, this approximation, this estimation that we did, this represents error. And that's okay. We're going to basically assume that if we just round down and follow the rules of rounding in math, we probably will come pretty close to the exact estimate for the number of repeats at this locus. And so there you have it. That's how we're going to interpret our chip results. So let's go ahead and actually run the chip for the 3 samples that I've been working on through the course of this experiment. So I'm up here in the DNA Learning Center's prep lab where we have our bioanalyzer instruments. So right over here we have the bioanalyzers themselves, and I have all the stuff that I need to carry out and run my chip laid out on the counter over here. We have this old computer over here that runs the bioanalyzer program, so it's a little ancient, but gets the job done, right? Let me take you on a little tour before we actually load the chip. And so this is the chip itself, and as you can see, it doesn't look like much. If you look carefully on the underside, you can see the microfluidic channels, although I don't think you can see them with this camera. But you'll notice a whole bunch of little holes in the chip. There's actually 12 locations for samples. There's also a location for ladder, kind of like the agarose gel electrophoresis, and we also have to put in a dye and a couple of other things. So that's the chip itself. This is a priming station that we use to actually push the gel, the actual gel mix, through the microfluidic channels. It uses vacuum pressure to do so. Over here, we have the bioanalyzer itself. So there are 2 instruments here, and they're both hooked up to this computer here. So if I open the bioanalyzer itself, inside you'll notice this little glass bead right there, which is the laser. And then over here, we have the actual electrodes that generate electric current that push the samples through the gel. So again, it's essentially working on the same principle as electrophoresis. It's just much more sensitive. So these pieces of equipment are pretty sensitive themselves, so we don't really want to mess with them too much. So let's go ahead and load the gel, or the chip, I should say. All of the reagents necessary to carry out the chip are located in this box here. And so what I'm going to do is pull out some gel matrix. Now, some of my colleagues have gone ahead and made the gel matrix for us already, which is very nice of them. So I'm going to go ahead and use that to load our chip here. Now notice it's basically like a blue solution, and it's very viscous, which you can't really tell, but it's gel. Remember, it's gel matrix, so it's kind of viscous. I've allowed it to equilibrate to room temperature, and I'm going to go ahead and load our chip, or put our chip into the chip priming station. That's this piece of equipment here, and it fits in very nicely right there. And now what I'm going to do is load 9 microliters of our gel matrix into the chip. So I'm basically going to follow the manufacturer's instructions on what to do and when to do it and for how long. So nothing really too mind-boggling or earth-shattering here. As a note, you should wear gloves when working with this stuff because there are some relatively noxious chemicals in here. And unfortunately, due to the current situation, I don't seem to be able to find any. So if you're doing this ever yourself, make sure you wear gloves. So I'm going to load the 9 microliters of our gel matrix into the appropriate hole in the chip. I'm going to do so carefully, and I'm going to try to avoid creating air bubbles, because again, air bubbles are kind of like the worst nightmare for this particular application. Once we have gone ahead and loaded that 9 microliters into the indicated well, I'm going to close the priming station. Okay, it snaps shut. Then what I'm going to do is push this little plunger down relatively slowly, and I'm going to push it until it goes right beneath this little clip here. There you go. Cool. Now I'm going to wait 60 seconds, and then I'm going to go ahead and release the clip. Okay, so it's been about 60 seconds. I'm going to go ahead and release this clip here, and as you can see, the plunger will go relatively back to where it was. It was actually at the 1 mil mark, and I'm going to make sure that it slowly gets back to that location as the instructions indicate. So I'm going to go ahead and pull it up to that 1 mil mark. Then I'm going to go ahead and open this and load another 9 microliters rather, of gel dye matrix into the indicated well. So I'm going to go back at my gel dye. And again, load 9 microliters into each of the indicated wells. Okay, 9 microliters of gel dye mix into each. Okay, now, before I move on, I'm going to double check and just make sure there are not too many bubbles going on in here, and it does seem like there are at least one. There is at least one bubble. Okay, and that seems to have worked itself out. And so the next step is to put 5 microliters of a marker into each of the wells. In fact, I'm going to put 5 microliters into all of the sample wells, as well as a well that is marked with a ladder. And that's literally going to be a ladder, just like we used in the agarose gel electrophoresis. So the marker is indicated by this little green dot here. And I'm going to put 5 microliters into all of the wells that I have not yet put anything into. So I'm going to go ahead and do this. It's not really, you're not missing out on much if you decide to skip this part. So I'm going to go ahead and do this real fast. Right, now that our marker is in, the next step is to add 1 microliter of ladder. Now, again, one of the advantages of the chip is that the amount of sample required is much less than it is in gel electrophoresis. So we're only going to need 1 microliter of sample as well when we get to that point. But for now, I'm just putting in the ladder. And the ladder is one of the most critical components of this whole process. If this is done incorrectly, or if the bioanalyzer reads a bubble when it's not supposed to, we could have some serious trouble because it will miscalculate where the ladder is supposed to be, and then all samples after that are going to be more or less lost. So this is a very important step. And at this point, I'm going to also double check and make sure that everything is nice and down into the wells, nothing up on the sides. Now I'm going to go ahead and load 1 microliter of each of my samples into the chip. So these are the 5 microliters of sample that we put aside at the very beginning of the part 2 of this experiment. So again, that's that little bit of sample that we saved in order to carry out this experiment. We could not do it after we added loading dye because the loading dye will unfortunately interfere with the chip and you will not be able to get good results. And at this point, also take note that I am changing my tips every single time. So I'm going to be loading both of my samples into sample 1 and 2. And then I'm going to load Christina's sample, that's my girlfriend's, into well number 3. Now, tricky thing here is if we don't have enough samples, so this chip can hold 12 samples. If we don't have enough samples, we have to put 1 microliter of distilled water into each one of the unused wells. So I'm going to go ahead and do that for the rest of the wells. You guys can catch up with me after. Okay, so we've added distilled water to each of the unused wells, so 1 microliter only. I'm going to go ahead and close this and put that away so it doesn't get damaged. All right, so the last thing I'm going to do is double check and make sure that I do not see any of these little micro bubbles that I was describing to you earlier that are the bane of this particular technique's existence. And I don't think I see any in there, but the last thing that I need to do is put our chip on the vortexer up here. And so I'm going to place this into the vortexer and I'm going to turn it on for about 60 seconds just to settle everything in place. Okay, 60 seconds is up. Now the chip should be ready to go. I'm going to check it one more time for those little micro bubbles that I was describing earlier. It does seem to be okay. So now I'm going to go ahead and place our chip into the bioanalyzer. So if you direct your attention up here, I am essentially going to just rest this chip right into the bioanalyzer so that it will fit right into those electrodes. So I'll close it up. The computer will then pretty much automatically register that there is a unit and it's detected that chip. You can see on the screen there, you can see a little image of the bioanalyzer chip right there. And I'm going to go ahead and just press start, and it's going to go ahead and do its thing. This takes a little bit of time, so in the meantime, let's learn about how our chip actually works. So while our chip runs, let's take a look at exactly what's happening. So I showed you this, and this is just a blown up image of the chip that I loaded moments ago. And let's go through this video that shows exactly what's happening. So if we take a look, or I should say a closer look at the chip, we can see the little microfluidic channels that I mentioned earlier. And what winds up happening is I went ahead and put that gel matrix, which we pressed down and forced it through all of those little microfluidic channels. And what winds up happening once the gel actually starts running, our samples, and we can see our samples actually coming right through here. And so our gel is, again, serving as that polysaccharide matrix in much the same way that the agarose gel did for the traditional gel electrophoresis, except this is a little bit more refined. It's a little bit more precise. So, but again, the same principle holds. The smaller the fragment, the quicker it's going to migrate through the gel. And so our bands will separate themselves out and they'll pass through a laser. That laser will then measure a certain amount of absorbance for each one of those fragments relative to the ladder, those reference markers, and generate that plot that I showed you earlier. And from that, we're able to calculate our size, exact size of our amplicons, and then by extension, the number of repeats at D1S80. Okay, so our chip is running successfully, it seems. I wanted to point out a few things to you here. So here's a summary of some of the data. And so this first piece right here is the ladder. And so if you take a closer look at the readout here, you'll notice that you've got a pretty even distribution of peaks, and that's good. That's what we expect to see. That means the data that we can see on our samples is going to be pretty reliable. The one thing I wanted to point out, however, is this peak right here. Notice it kind of goes below that sort of baseline there. That's probably a bubble. I guess I wasn't as good as at getting rid of some of those little micro bubbles as I thought. Oh well. But if we take a look at the samples, what we see here, importantly, is our ladders, our markers here and here on either end. And so the size of the base pairs here, it tells you 15 base pair at the minimum end and 1500 at the max end. So that's exactly what we hoped to see. There's a lot of noise going on in the background here, and that, look, so for instance, there's a secondary peak over here, and that's not supposed to be there. So once this is all said and done, I'm going to go ahead and mark the indicated peaks that I think are reliable peaks based on the gel electrophoresis results. That's another good reason to do the agarose gel as well. You can sort of cross-reference with the agarose gel. And by the way, the chip does its own little gel electrophoresis as well. And you can tell right up here, it's indicating that here's the ladder, here's the other ladder, here's that background, and then here's two peaks right here. And that's basically what we saw on the agarose gel. So this unit essentially goes through and sequentially builds each one of these plots. And so this is my second sample, and I see another bubble here. Again, I guess it's a little more messy than I thought, but whatever. And then over here, I see my two peaks that correspond to my actual amplicons that I'm interested in. And right off the bat, I'm noticing that the peak that it's assigned for this taller one here is the same as the last one. So I think we're in good shape here. It did not indicate a peak over here, but that's okay. I'm going to go ahead and do that, and we'll talk about it in a second. So let's let this finish its process here, and we'll take it from there. All righty, so if we take a look at the chip results, I've got them all pulled from the Bioanalyzer, and I have them summarized on this slide here. In comparison to our gel results, we can see the differences, and so and the similarities for that matter. So the good news is, as expected, because those first two samples, remember the Jeff samples. Remember, I went through a DNA extraction with you guys, and then I had previously done a DNA extraction the same way. And so both of my samples on the gel appear to be about the same, and both of my results on the chip confirm this. So if you notice, all the way on the bottom left here, we had two peaks between the ladders. We had 533 and 609 for the first sample, and 531 and 611 for the second sample. Going through the calculations that I just showed you a little while ago, we come up with the genotype of 2429, 2429 in both cases. That's awesome. That's exactly what we would hope for. Now, the really, really cool thing about the chip is if you take a look at Christina's sample, notice that she has a couple of peaks as well. She has two peaks, 544 and 606. Now, unfortunately, as I was mentioning to you a couple of moments ago, these weird peaks in the middle here, those are bubbles. And same thing goes for my second sample over here. Now, those contribute error, and they wind up pushing the absorbance peaks around a little bit. So there might be a little bit of error in these estimates. And if you go through the calculations, you'll see that there's just a little bit of rounding that you have to do. But that's okay. These results are good enough, I think, to really draw conclusions about the chip. And I said earlier that if you remember on the electrophoresis gel up here, I said that I thought it looked as though one of her bands might be a little higher than mine. But the second band here, that top band, it looks like it's in the same exact location. And if you go through the calculations, you'll see that she comes up with 29 for one of her alleles and 25 for one of her alleles. That's crazy. So even though we kind of thought they could be the same genotypes, we could be the same genotypes based on the results up here, they're inconclusive. But when we compare them to the chip results, we see that she has one repeat fewer than I do, apparently. She'll be very happy to hear this, I'm sure. So that's pretty interesting. And so we can see just how interesting the difference is in terms of precision the chip is in comparison to your traditional gel electrophoresis. Now, how rare exactly is this genotype? You know, how 24, 29, and 25, 29. So in order to do this, we have to compare this to the total population. And aside from going out and doing this testing all on our own, we have to find a ready available database that we could compare to. And it just so happens that Yale has come up with a database for some of these markers, and it's called Alfred. So if you go to Google, you can simply search Alfred, and we're going to type in D1S80. So if you just simply go to Google and type in Alfred genotype frequency or something along those lines, you'll come up with the allele frequency database. That's what Alfred stands for. And so this database has a lot of different genetic information about polymorphisms and frequencies of their occurrence in a population based on ethnicity. So we're interested in D1S80, so let's go ahead and type D1S80 in. I'm going to click Go. And we're interested in the top one here, so we're going to go ahead and click that. And then we'll have the sort of this summary page here. We are going to click on this indication right here. And this will bring us to a frequency, or the possibility to come up with a frequency table. So if I click the frequency table, it will summarize all of the frequencies of each different repeat in a population that they've sampled. So if you notice, here's our frequency table here. Across the top here, we have a summary of all the different numbers of repeats that are out there. So again, we're going from 14 roughly to 42 more or less. There are some oddball ones out there, which I didn't tell you about earlier, but that's okay. Just know that they do exist. But typically we're dealing with between 14 and 42, as you can see. So let's take a look at those genotypes that we had. So here we had a 24 and 29. So here's my allele 24. That's the 24 allele. So let's go ahead and go down, and it cross-references ethnicity over here on the left. So let's say that let's go with my Italian heritage. So here's my Italian heritage down here underneath Europe. And so let's see, we'll follow the column all the way down. And here we go. So this tells us, now these are two different sample populations here of quote unquote Italians, but it seems like a lot of Italians have this particular allele frequency. So 43% and 37%. Or, I'm sorry, they have this particular allele rather. What about 29 on the other hand? So if we go over a little bit and follow all the way down to the 29 allele column, over here by Italians, oh wow, look at that. So there are many fewer individuals in an Italian population that have the 29 allele. So this is a good way to tell exactly what the probability is of finding someone with my particular allele, with my particular genotype. So you would just basically take the probability of getting one allele in a population, and you would multiply it by the probability of getting that allele, getting the other allele, and given your ethnic background. And that would give you a rough and ready estimate of the probability of encountering that allele combination in a population that's representative of your ethnicity. Now, if you're a criminal investigator, you may want to use multiple alleles. And so each one of these probabilities will compound. And you want to be sure of a particular conclusion that you can draw about these allele frequencies. So that's why criminal investigators will investigate many different types of alleles. And so this all has history, by the way, in a scientist named Jeffry, who developed a technique known as Jeffry's probes, using these things called mini satellites to identify individuals on the basis of DNA. And so this was sort of the first vestiges of these DNA profiling. And this is not just necessarily useful in forensics, it can be useful in paternity testing, immigration tests, and so on. So there are a lot of applications of this idea of DNA profiling. And like I said, in forensics specifically, when you're dealing with essentially a life or death situation for potentially convicting someone, sending them to prison for possibly for life for a crime. You have to be, the FBI has to be really sure, and other law enforcement agencies have to be really sure that the information that they're getting, and it has to be highly unlikely for a particular DNA profile to be found at a crime scene in order for them to draw serious conclusions. And so there are a number of different markers that law enforcement agencies will use. And so CODIS is a criminal database of DNA information, and it incorporates information about various different markers. And so they're kind of, some of them are summarized over here on the right. And the idea is they're just taking, to establish a really, really clear DNA profile, they're using information from multiple different marker locations. And this is pretty profound too, because in forensic investigations, DNA evidence has become very important. It's become a norm in cases throughout the country and throughout the world. And so it's a very important forensic technique because it really can carry quite a bit of clout in a legal situation. I hope you've enjoyed the forensic DNA profiling lab. So as we saw, we figured out our exact number of repeats, our exact genotype at the D1S80 locus in much the same way that a criminal forensic investigator might do so for a suspect. We took a look at several different techniques for visualizing our DNA, including gel electrophoresis and our DNA chip. And I hope you had a great time watching this video. I hope you learned a lot. And when you guys come to the Learning Center, I encourage you to get excited about this lab. This is one of my favorite experiments that we do here. And I hope to see you guys soon. Have a wonderful week, and we will see you next time. Check out DNALC Live for more excellent content, and stay safe.